Hi everyone, today we're checking out TAME-VS, a powerful Python tool that brings an end-to-end approach to ligand-based virtual screening in drug discovery. The tool takes a UniProt protein ID, expands it with similar targets, pulls in known bioactive compounds, and then trains ML models like random forests and neural networks to predict new hits. It's open source, flexible, and designed to make early-stage drug discovery faster, cheaper, and more accessible to scientists. The pipeline runs through several key steps: target expansion, where similar proteins to the query are identified; compound retrieval, which collects active and inactive molecules; vectorization, where molecular fingerprints are computed; machine learning model training, using random forests and multi-layer perceptrons; virtual screening applied to a user-selected chemical library; post-analysis, where drug likeness and key properties are evaluated; and finally, data processing, which outputs a prioritized hit list along with summary reports. Importantly, users can enter the pipeline at different stages depending on the data they already have, and numerous parameters in the shell and Python scripts can be easily tuned to fit specific project needs, making the tool both comprehensive and highly adaptable. First, the method starts with target expansion. You provide a UniProt ID, and the tool runs a blast search to find proteins with similar sequences. By default, it uses a 40% sequence identity cutoff. This is reasonable for finding homologs, but this value may also exclude some related, biologically relevant proteins. The good news is that users can change this threshold. The next step is compound retrieval. The tool connects to the ChemBL database to collect active and inactive compounds for the expanded protein set. This is a clever way to build a training dataset quickly, but it's highly dependent on ChemBL coverage. For well-studied targets, you'll find thousands of compounds. For some novel targets, you might find almost nothing. If that happens, you could bring in your own in-house data or explore other methods. Once compounds are collected, the vectorization step turns compounds into numerical fingerprints. Various fingerprint types are available, including the well-known circular Morgan fingerprint. A basic compound curation step such as removing salts could be beneficial at this stage. Now comes model training. The tool builds two classifiers using random forests and multi-layer perceptrons. These are reliable and fast for this kind of data sets. Other machine learning methods could be added, which could eventually improve accuracy and generalization. Trained models are then used for virtual screening. By default, the method screens the enamine diversity 50K library, but users can also plug in any chemical collection. As it stands, the pipeline isn't set up to handle millions of compounds, but since the code is open and modular, it could be extended to scale up to much larger libraries if needed. After screening, post-analysis evaluates drug likeness and some key properties like molecular weight, log P, or hydrogen bond counts. Finally, the data processing step compiles everything: the top 1% of predicted hits, summary reports, and performance metrics. This end-to-end pipeline is a real strength of the method. However, like any automated pipeline, it can only be as good as the data going in. If target homologs are sparse or ChemBL data is missing, the output will be limited. Still, the modular design means you can plug in other models, richer data, or new compounds. At the end of the pipeline, the tool brings everything together. Both the random forest and the MLP models produce their own rankings of compounds, but instead of relying on just one, the platform also computes an ensemble ranking. This is done by averaging the ranks from both models, so molecules that score well across methods rise to the top. The final output includes the individual model scores, the ensemble ranking, physicochemical properties, and a filtered top hit list. To try the tool, I ran the pipeline on a serine protease target. I made a few modifications to the scripts to better fit my project, and the workflow ran smoothly from target expansion to final hit selection. The output included a rank list of candidate compounds with physicochemical properties and QED values. And here I'm showing only 2 molecules. One can sort the compounds using various parameters, and I could also use a larger collection as input for the virtual screening. Experimental validation would be needed, but it's a great example of how the tool can quickly generate a prioritized hit list for further exploration. At this stage, in a real-world scenario, significant work would still be needed to further analyze the selected molecules, evaluate the performance of the trained models, examine the datasets used, and explore additional methods such as molecular docking if a 3D structure of the target is available. This is it for today. I hope you found it useful and inspiring for your own projects, and until next time, take care.